Okay, so we mentioned how primary neurulation occurs. We talk about the signals that control the formation of the hinge point, but let's have a look on what's happened in secondary neurulation. In secondary neurulation, we have that a group of mesenchymal cells that are both ectodermal and mesodermal in origin condense with each other to form a medullary cord. A medullary cord. This medullary cord is formed just below the ectoderm and above the notochord. The medullary cord then undergo a process of cavitation, forming different cavities, and different cavities then collapse together, forming a single lumen. When this lumen is formed, then we have when this is become the neural tube. Notice that the ectodermal cells that form secondary neurulation express high level of SOX2, and also the surface ectoderm and the neural ectoderm in secondary neurulation are completely uncoupled. They develop independently. They don't interact with each other. Differently this compared to primary neurulation. In primary neurulation, the two group of cells were actually attached to each other, and they need to detach from each other during the process of neurulation. Okay, so now the neural tube is formed, but inside the neural tube, we have the formation almost immediately of a dorsoventral gradient of expression. As I mentioned a minute ago, the notochord is expressing SHH. SHH expression from the notochord induced the floor of the neural tube to also express SHH, while the external ectoderm, the external epidermis, if you want, express high level of BMP. And this high level of BMP induced the dorsal part of the neural tube cells also to express high level of BMP. So now in the neural tube, we have that the dorsal part of the neural tube, so the dorsal cells are expressing BMP, or if you want, TGF-beta signaling, which is the same. While the floor, the ventral part of this neural tube are expressing SHH. And therefore, these proteins form a gradient, diffuse along the neural tube, and form a gradient. And we have a dorsal to ventral gradient of TGF-beta signaling, and we have a ventral to dorsal gradient of SHH. Now, why this is important? Well, this is important because different kinds of neurons require for their formation different level of SHH and TGF-beta signaling. So, for example, high level of SHH specify V3 neurons, which are near the floor, while interneurons, for example, require high level of TGF-beta. And some other neurons require intermediate level of SHH and TGF-beta. So what's happened is that different kinds of neurons are now induced and formed along the dorsal ventral axis in the neural tube because of the gradient of SHH and TGF-beta. So once again, gradients of proteins, in this case SHH and TGF-beta, allow the cells to understand the grid where they are in a three-dimensional space, and therefore to express the correct pattern of genes to give a different structure in the correct place and time during development. So we already saw a few times that gradient of expression, an anterior-posterior gradient of expression, allow the cells to understand their position, if you want, in the along the anterior-posterior axis, so they can do different things. And now we have another gradient in the neural tube that do the same job. Basically, gradient of expression, gradient of proteins, works like, if you want, GPS. Depending on the concentration of different proteins, the cell will be doing different things. Okay, so hopefully you will be able to explain the formation of the anterior-posterior axis. You will be able to explain, after revising this lesson, the role of different genes that are involved in the formation of the anterior-posterior axis, their expression and their proteins. You should be able also to you should be able also to discuss and explain the formation of the neural tube. So here we are talking about primary and secondary neurulation, as well as junctional neurulation. What genes are there? They are expressed. What's the role of the notochord, and why there is a gradient, a dorsoventral gradient in the neural tube when the neural tube is formed.